going to start recording our session for today. Okay, before I show you our invited speaker, let me introduce him to you. He graduated from our University of the Philippines, Los Baños, taking up Bachelor of Science in Biology, major in Microbiology, cum laude last 2011. And he took his graduate studies also in the same university, taking up Master of Science in Microbiology, minor in Veterinary Medicine last 2015. And he was able to join the Department of Veterinary Paraclinical Sciences, College of Veterinary Medicine as a University Research Associate 2 last 2014, and was able to join again with the same role from 2018 to present. And I am privileged enough to have worked with him in our project, College of Veterinary Medicine, PF 480, under the Department of Veterinary Clinical Sciences, working on the profiling of economically important diseases of swine and cattle in the Philippines, for enhanced disease management, surveillance, and control. He has other researches as well, such as rapid diagnostics and control strategies for enteric bacterial pathogens in backyard and commercial poultry production in Thailand and the Philippines. And he is also part now of the UPLB program for zoonotic diseases. Without further ado, let me introduce to you Mr. Sobel Ezra A. Salamat. Let's give him a round of applause, please. Yan. Okay. Good afternoon, sir. Thank you for joining us. And the topic for today is on the polymerase chain reaction and its applications. You may have the...
Good afternoon, Doc. Good afternoon, everyone. Thank you for the introduction.
Good afternoon po.
Okay, sir, you may now have the screen.
Okay, I'll share the screen. So, pwede ko pa-enable.
Okay, sorry. Okay. Okay, enabled na po.
Sige, Doc. May kita po, Doc?
Yes po.
Hello po to everyone. So, our lecture for today is about the introduction to PCR and its applications to veterinary research. So, as mentioned by Doc Maying, I'm a University Research Associate 2 at the College of VMED, UP Los Baños, and my current specialization is on molecular microbiology. So, feel free to interrupt me anytime in case you have any clarification. Pero yung mga questions siguro, we can reserve it for later. But if you have any clarifications, feel free to interrupt me anytime. Okay, so there are 4 parts for this lecture. So first would be nucleic acid isolation. Then we'll talk about conventional or endpoint PCR. Then we'll talk about real-time PCR. And for the last part, we'll look into the applications of PCR and real-time PCR. Okay, so for the first part, so we'll start with nucleic acid isolation. So just a review. So the genetic material of organisms can be DNA or RNA. So DNA is usually for eukaryotes and bacteria, and while the RNA is usually reserved for some viruses. So DNA is usually double-stranded, and RNA is usually single-stranded. So you'll see a lot of these later. Okay, so review lang. And the DNA is composed of in general, 4 nucleotides. So cytosine, guanine, adenine, and thymine. While for the RNA, it's almost same except that thymine is replaced by uracil. Next, nucleic acid isolation. So for any molecular biology technique that you do, almost always, not all the time, but almost always, you need to do nucleic acid isolation first because you need the DNA or the RNA, and usually they're inside the organism or the virus, so you need to take them out. So the preceding step in almost any molecular biology technique would be DNA or RNA isolation. So it can be performed using a variety of conventional organic solvents or commercially available isolation kits, as you can see. Kita ba yung mouse ko or hindi?
Kita po yung
Kita
map.
siya? Kita siya. Okay, good. So here is a couple of the nucleic acid isolation kits. So there are a lot of brands available in the market with different applications. So all nucleic acid isolation kits are different, but generally, not always, but generally they have 4 general steps. So first would be cell lysis. So this involves physical or chemical disruption of the cellular protective structures of the bacteria, animal cell, or the virus. And its purpose is to release the nucleic acid. So as you can see here, for bacteria, the DNA is localized in what is termed as the nucleosome or nuclear zone within the cytoplasm. So for the animals, meron pa silang nuclear envelope. But the point is the DNA is inside the bacteria, the animal cell, or the virus, and this needs to be taken out before you do any analysis. Okay, so usually you can do chemical cell lysis. For example, you use detergents or enzymes such as this one, or you can use mechanical disruptions such as this one. So this is using beads. So this is using beads to bash the cells in order to disrupt and break apart the cellular membrane. Okay. So next, after you release the nucleic acids, you need to capture them or separate them from the rest of the cellular components or the rest of the samples that you don't need. So the second step is nucleic acid capture, where the DNA or RNA are captured. So this may involve using silica membranes, such as this one. So if you can see here, this is a spin column. The white part is the silica membrane, which has a natural affinity for DNA and RNA. So para siyang magnet. So dyan dumidikit yung DNA or RNA. And you can also do magnetic particle certification, magnetic particle separation, which we will not discuss because it is this is just an introduction. But it can be also used as well. And you can also use centrifugation. So centrifugation is usually employed for larger volumes of nucleic acid, such as when you isolate DNA from cultures, bacterial cultures, or plant cells. So these are the different methods of nucleic acid capture. Next would be washing or cleaning. So of course, after you've captured the nucleic acids, it's not clean. So you need to remove the debris, you need to remove the other inorganic and organic substances that are not nucleic acids. So that includes fats, that includes proteins, that includes other chemicals that you don't need for your analysis. So you need to remove those impurities using a variety of buffers. So for example, for a genomic isolation kit, you will almost always see that there are wash buffers. Usually there are two or three of them. So they will be able to wash the non-polar and polar impurities inside that is still clinging onto the nucleic acids. And also, you need to remove the wash buffer after you wash the DNA or RNA. So after washing or cleaning, you will elute or isolate the nucleic acid from, example, the spin column. So after cleaning, the DNA or RNA is still here. So you need to remove it in order to use it. So you elute. So usually this is usually done by dropping water or water-based buffer because DNA and RNA is soluble to water. So usually you drop that buffer or water here, then you centrifuge, then you get the DNA, pure DNA or pure RNA. So those are the 4 general steps for a lot of nucleic acid isolation procedures. Okay, next, So don't be overwhelmed. So this is just a chart showing the different DNA and RNA isolation kits available in the market. So you have different brands here. You have different purposes. So the point of this table is just to show you that different kits have different purposes, and they also have different types. For example, this Kia-Amp DNA mini kit from QIAGEN can be used manually, VMEM, or using automated methods. So it uses spin column and vacuum method. So, and it can be used for blood, cell-free body fluid, cell tissue swab, for DNA. So note, kaya siya DNA kit because it cannot be used for RNA. Same here, viral RNA kit, you can only use it to isolate RNA. You cannot use it to isolate DNA. So same thing. Meron naman na you can use for DNA and RNA, such as, for example, mag-attract blood DNA RNA isolation kit. This can only be used with automated methods. So note, if you don't have the machine, you cannot use this because it is for automated protocols only. And it uses magnetic particle isolation method, and it can only be used for blood, and it isolates both DNA and RNA. So the main takeaway here is that when you graduate and you work in a laboratory and you do DNA RNA isolation, you need to be careful in what you purchase because there are a lot of kits available in the market and each of them have their own purposes. So yun lang yung takeaway dito sa table. Okay, next part would be conventional PCR. So this is the most, I think the most common PCR method that's being used, and likely the first kind of PCR that you will be learning in the laboratory. Review lang ulit. So DNA is the information storage of organisms, and they have different base pairings that are very specific. So A pairs to G, G pairs to C, T pairs to H, C pairs to G. So this will be very important later sa PCR. So what is PCR? PCR is a laboratory technique where a segment of DNA is artificially and selectively copied exponentially. So it's like photocopying a book. So when you photocopy a book, you don't usually photocopy the entire thing. You photocopy only a page or a few pages. So it is the same thing. In PCR, you don't usually copy the entire genome because that's too big. You only copy a segment, maybe a few hundreds or a few thousand spaces long. So the purpose of that is to generate millions to billions of copies in order to know if that segment is present. So for example, you have one copy, then after one cycle it becomes two. After one cycle, another cycle you have 4 copies, another cycle you have 8, 16, 32, until you have millions to billions of copies. So as mentioned, it's a chain reaction comprised of several repeating steps, which we will look into later. So the end goal is to target and detect specific DNA regions by selectively copying them. Okay. So this is better explained in an animation. So I will be referring to a video by Shomo's Biology. You can also watch him on YouTube because this explains the steps of the PCR process. So as mentioned earlier, the required precursor of the polymerase chain reaction would be nucleic acid. So you cannot perform PCR without isolated DNA. There are exceptions, but usually you need to isolate the DNA or RNA first. Okay, and you also need kits, or if you're using organic solvents, you need the organic solvents first. Okay, so let's watch the animation. Rinig ba? Okay, okay.
No sound, sir, pero we can view the animation.
No sound.
No sound.
Wait lang. Bakit kaya? Anyway, explain ko na lang. So if you can see here, that's the entire genome, but you're only copying small segment. So that's your target region. So you'll need several other components, polymerase, the nucleotides or the building blocks of the DNA, and also the primers, which we will discuss later. But these primers, the purpose of this is to target the specific regions of the DNA that you will be amplifying. So this DNA polymerase is usually obtained from hot springs. So this is from the bacteria Thermus aquaticus. So the first step is to denature the DNA, and that would require higher temperature. So usually 95 degrees Celsius, and that would result to the strands of the DNA separating into two. So that's the forward strand and that's the reverse strand. So you need to separate them because you need to attach the primers. So afterwards, you need to reduce the temperature down to the annealing temperature, which is usually between 50 to 60 degrees. Then what will happen is that the primers will be attracted to the specific region of the DNA where it's complementary. So primer binding is very specific. So this will only bind to this region and not in any other region, as you will be able to see later. Afterwards, temperature is raised to 72 degrees Celsius, which is the active temperature of the Pac polymerase. And what it will do is it will add the building blocks of the DNA one by one. So you see that one strand will become two strands. After one cycle, you have two copies of DNA from one copy. Then you will repeat the cycle for usually 25 to 40 times. So after the second cycle, you have 4 copies of target DNA from 2 cycles at cycle number 1. After cycle 3, you have 8 copies. 2 of them have the target DNA, while some of them have these extensions. This is important to note because these long DNA segments that have the target DNA but sometimes have part of the genomic DNA can interfere with electrophoresis later. So sometimes you'll see this as a band, smear. So it's very important to optimize your reaction so that you'll only get a lot of this target DNA. So this is the only thing that you need. So as much as possible, you have very few of these, but a lot of these target DNA. So after each cycle, the number of copies of those fragments that only have your target sequences will increase. So after 25 cycles, you'll have approximately 33 million copies of DNA. So this copies you will be able to visualize in your gel electrophoresis. Okay, so that's the end of the animation. Oops, next slide. So these are the components, PCR core component of the PCR reaction. So first you have the template DNA, so that's your target. So usually you get them from your bacteria, from your virus, from your animal, etc. Then you have your primer pair that binds to both ends of the target DNA region. We'll take a look at these primer pairs later. Then you have the thermostable DNA polymerase that copies the target region. So this is the one responsible for copying the DNA. Next, you'll have the deoxyribonucleotide triphosphates, the NTPs, which are the building blocks of the DNA. And next, you'll have the buffer, which contains various ions that will stabilize the PCR reaction. So these are the PCR core equipment. This is the bare minimum that you'll need. So you have your thermal cycler. As you saw in the animation earlier, you need at least 3 temperatures. So the denaturation temperature, the annealing temperature, and also the extension temperature. So that the thermal cycler will be the one who will be performing all those thermal cycles. So this is just a very fancy hot plate. So it changes your temperatures depending on your need. Next will be your gel electrophoresis, which will apply a voltage to a gel with your DNA so that you'll be able to separate them. We'll take a look at that later. And also your gel documentation system, which will be your camera that will record the DNA bands. So this is an animation of the PCR primer specificity. As I mentioned earlier, PCR primers are very specific in terms of the regions where they will bind. So for example, this is your template DNA. Then you will denature them. Let's try inserting a wrong primer. So you can see they won't bind because it's not specific. It's not complementary. C is complementary to G, as you can see here, but C is not complementary to C. A is not complementary to A, but A is complementary to T. As long as there are a lot of non-complementary regions, then this will not bind. So this will be rejected by the template strand. However, if we try to add a correct primer, a complementary primer, then the two will bind. So this will bind to the 5' to 3' strand, and this will bind to the 3' to 5' strand. Then afterwards, DNA polymerase will be detecting this 3' overhang. So once the DNA polymerase, the polymerase will sense the 3' overhang, it will proceed to polymerization. So that's how the primers work. So they're very specific. Next, after you do your PCR in the thermal cycler, you need to do gel electrophoresis. Because as you can see earlier, the PCR reaction is a mixture of your template and your amplicon. So you need to separate them. So it's a laboratory technique where DNA fragments, usually from PCR, are separated. So you pass an electric current anywhere between 110 to 120 volts across the gel containing the DNA. So this composed of agarose, the gel, and you have wells where you drop the DNA, and afterwards you apply an electric current from anywhere between 20 to 45 minutes. Then separation is usually based from the size. Then this will accurately determine if the PCR is positive. So the negatively charged DNA molecules move towards the positive electron. Okay, so they will move from here to here. Okay, so electrophoresis is like threading a thread through a cloth. So if you have tried sewing before, you'll know this. Larger fragments travel through the gel slowly. So this is a microscopic view of the gel. As you can see, it's like a web. So larger fragments will have a hard time passing through this web, while smaller fragments will have an easier time traveling through this web. So if you have a gel, then you subject this to electrophoresis. The DNA samples that are the smallest, like this one, will travel faster, while the larger ones will travel slower. And you have a DNA ladder with predetermined sizes of DNA fragments, and this is how you will measure. So the ladder is like your ruler, and you will match the size of the bands. So if this is 100 base pairs and this is 200 base pairs, then you can assume that the size of this band is somewhere around 140 to 150 base pairs. So that's how you measure using gel electrophoresis. But then again, you can't see the DNA. So what you do is to stain the gel. So you stain the gel using a fluorescent DNA binding dye. So these dyes fluoresce or glow when bound to DNA and exposed to UV light. So you will encounter chemicals like ethidium bromide or gel red. So as you can see, this is the DNA. This is the one strand, and this is another strand. And this is ethidium bromide. So what ethidium bromide does, it is binds between the two strands and it will fluoresce or glow under UV light. Okay, so this is an example. So in gel staining, millions of copies of DNA that are generated by PCR, which are identically sized, will cluster to one band kasi same size nga sila. And it will fluoresce. So for example, if you use different dyes, so there are a lot of dyes that are out there. If you use diamond dye from Promega, it will fluoresce green under UV light. And if you use gel red, for example, from Biotium, you will get a red fluorescence. And if you use ethidium bromide, you will also get orange red glow. So as you can see here, this is a 100 base pair dye. So the size of this band is 100 base pairs. So this 100, 200, 300, 400, 500, 600, 700. The size of this band is 651. So this is the positive control, and these are the samples. So you can see that the positive control is actually 651 base pairs because it is between the 600 band and the 700 band. So sample 1, 2, 3, 4, 5, 6, 7 are positive. Sample 8 is negative. Sample 9 is a negative control, so it should be negative. Sample 10 is the positive control, so it should be positive. Okay, so same here. Sample 1 is negative because although it has a band, its size is not 651 base pairs. Sample 2 is negative, sample 3 is positive, sample 4 is negative, sample 5 is positive. Okay, so it's that simple. Next, we go to real-time PCR. So this is the more advanced version of PCR. So here you don't need the gel. That's why it's called real-time PCR because while it's running in real time, you can know whether or not your sample is positive. So a real-time PCR, unlike a conventional PCR, is a type of PCR where the amplicons are detected in real time, as I mentioned earlier, as the reaction progresses. It utilizes fluorescent DNA binding dyes or fluorescent DNA probes to detect amplicons. So these are the samples of the PCR, the real-time PCR machines that you will be seeing in the laboratory. So there are two types of PCR. The first one is DNA dye-based real-time PCR. So they use fluorescent intercalating dyes that bind to amplify double-stranded DNA. So something like this. Similar to ethidium bromide, they will bind to between the two strands of the DNA. They are relatively cheap. For example, cybergreen. These fluorescent DNA binding dyes are non-specific. So whatever size of your amplicons, they will bind. As you can see here, it will bind to this amplicon. It will also bind to this amplicon. It will also bind to this amplicon. So it will not matter what is the size of the amplicon, as long as it's double-stranded, it will bind. So that's why it's not as accurate, but it's cheaper. So these kits are also limited in a way that they can only detect one target per reaction. So you cannot detect multiple targets in one reaction. So this is the cyber assay or DNA-based qPCR. So the special thing about these dyes is they are fluorescent. So they are weakly fluorescent when there is no double-stranded DNA. But once they bind to the DNA, double-stranded DNA, they will fluoresce intensely. So if you will see, during the course of the PCR reaction, during the denaturation phase, the fluorescence is weak. Then during the annealing and extension phase, the fluorescence will slowly increase. Then once the reaction is complete, the fluorescence will be at its most intense. So this is how you will detect it. So on your first cycle, you only have a few copies. Then on the second cycle, you have 2, you have 8, you have 16, until you have millions of copies. So since the dyes bind to the double-stranded DNA, there will be more double-stranded DNAs that the fluorescent DNA binding dye will bind on. So as you progress through the cycles, there's a relatively increase in the fluorescence of the reaction. So that's how you will be detecting whether or not the qPCR reaction is positive or negative. So if there's amplification, if there's a signal, then it's likely positive. Otherwise, it's negative. Okay, next is the fluorescent reporter probe-based kits, or in short, the probe-based qPCR. So these kits rely on reporter probes that work in concert with the primers and DNA polymerase. So the probes are tagged with a fluorescent-based dye and a quencher on the other side. Then these probe-based kits are relatively more expensive, somewhere between 2 to 3 times more expensive than the cyber kits. This is primarily due to the additional quencher. These quenchers are usually very expensive, as much as anywhere between 20 to 40,000 pesos per vial. So they are very expensive. However, probe-based kits are more specific, as the probe will only bind to its target sequence. So since this is a probe similar to a primer, it will be very specific to the sequence, and thus this kind of reaction is more specific and more accurate. So these kits can also do multiplex assays. For example, you want to detect, what's a good example? PED and TGE. So if you want to detect both PED and TGE, if you use a cybergreen assay, you need to use 2 tubes per sample. But if you use probe-based PCR, you can detect both TGE and PED in just 1 tube, because you'll just use 2 probes. So you can do as much as 5 or 9. I've seen publications using detecting having multiplex PCR reaction as high as 5 or 9. So that's the advantage of the real-time PCR using probe-based method. Okay, so this is an illustration of the probe. So this is your probe. For the conventional PCR and the cybergreen PCR, you only have 2 primers. But here you have quote unquote 3. One of them is the probe. So the other 2 are regular primers, and your probe will have a fluorophore, which is a fluorescent dye on one end, and you have a quencher on one end. So the job of the quencher is to absorb the light coming from the fluorophore. So if you look at this probe from afar, you cannot see any light, because this is emitting light and this is absorbing light. And this one is close to this one. So it will not emit any net signal. But if this is your target and the forward, the reverse primer, and the probe all bind to the target, then what will happen is the polymerase will have the polymerase has a 5' to 3' exonuclease activity. What that means is that if it detects, it runs over a strand of DNA, it will digest it. So once it moves from the left to the right, it will encounter the oligonucleotide probe and it will digest it. So what will happen is that the probe, the fluorophore will separate from the quencher. So maglalayo sila. What will happen is that the quencher can no longer absorb the light that is being emitted by the fluorophore, and therefore you can get a signal. So that is the how the detection method of the Tachman assay works. So for example, if these 2 primers bind to the target, but this does not bind. So what will happen is that the polymerase cannot digest the probe. So we will get a negative signal. Okay. So you'll detect this using real-time PCR. So real-time PCR usually have a light source. Then they will shine that light source on your PCR sample. Then all the positive samples will fluoresce, and that will be detected by the fluorometer. So this is what your graph will look like. So it's an exponential graph. So if you see an amplification, then it's likely positive. Okay. So there are a lot of qPCR dye. So this is the reason why you can do a lot of, you can detect several targets in a reaction. So if you want, you can assign PED in the blue channel, and you can assign TGE in the green channel. So if you see only blue signal, you're positive for PED. If you only see green signal, then it's positive for TGE, and so on and so forth. So I think they have 6 or 7. I think then there are other dyes that do not emit UV light, visible light. So they are either on the UV, somewhere in the UV aspect. Okay, so the point is there are different dyes that you can use. Okay, so let's go to the applications. What can you do with PCR and real-time PCR? So first is pathogen detection. So as I mentioned earlier, the primers used in PCR and real-time PCR are very specific to DNA. And since different organisms have different sequences, you can use that to your advantage. So for example, this is a sequence from Haemophilus parasuis 16SR DNA. It's different from the sequence of PED, spike in flag. So what you need to do is to design a primer specific to this sequence, so that after you add them to your PCR reaction, you will only detect PED. Or if you want to detect Haemophilus or Staphylococcus or Streptococcus, you find the sequence, you create a primer, then afterwards you will be able to use that primer in a PCR reaction. Right? So for pathogen detection, it has a lot of applications. For example, disease diagnostics for your patients, admitted patients, or for farm animals, for example. You can also do disease monitoring to, for example, ASF. If you want to know whether or not a farm has ASF, you can use PCR or real-time PCR. You can also do evaluation of antibiotics or antivirals. You can, for example, antibiotic resistance is conferred by genes, usually plasmids. So if you want to know whether or not certain bacterial species has antibiotic resistance, you can do the Kirby-Bauer method, or the one in the phenotypic analysis, or you can also do PCR. Okay. So next would be evaluation of antiseptics or disinfectants. If you want to know whether or not you have killed the bacteria or the virus, then you can also use PCR. And also for vaccine evaluation, you can also do PCR. Okay. Next, for vaccination, you can do PCR to properly administer vaccine. So let's say a theoretical scenario wherein, okay, so for example, infectious bronchitis. So the infectious bronchitis virus has several strains. And also it has several vaccine strains available in the market. For example, you have Massachusetts strains, you have H120, etc. So I have a hypothetical scenario wherein there's an infectious bronchitis outbreak in region 4. Then your poultry farm is in Laguna, is not infected, but it's scheduled for vaccination. So because Cavite is near Laguna, then it's likely, it's possible that your flock will eventually get infected. Then the poultry farm in Cavite, which is positive, was tested, and it was identified to be a Holland strain because of PCR. So what you'll do is to administer H120 IV vaccine for maximum protection. Because if you vaccinate with a heterologous strain, for example, if you vaccinate a Massachusetts strain, you might get only 80% instead of 100% or 95%. So you'll also see that here in the current situation of the pandemic, they're talking about boosters. That's why they are not yet advising vaccination with boosters that are not based on the Delta variant, because it might not be as effective to the Delta variant as to compared to a vaccine that is engineered against the Delta variant. Okay, so that's why PCR can be used as one of the tools, not just the only tool, but one of the tools for more effective vaccination. Next would be gene expression. So if you're doing studies in physiology, for example, then if you want to measure proteins, for example, cytokines, you can detect the protein, you can measure the protein, that's possible, that's usually very difficult and very expensive. So what you can do instead is to do gene expression analysis. Or instead of measuring the protein, you measure the precursor, which is the mRNA. So theoretically, if you have higher mRNA expression, you have higher protein expression. So instead of measuring this, you measure the transcript or the mRNA. So for example, some applications would be cytokines. So for example, you're studying the progress of a disease. How does a certain pathogen activate the immune system? What are the immune cells involved? So one of the tools that you can use would be cytokine analysis. So you analyze cytokine expression to measure immune response in response to infection. Another one is if you will be detecting virulence genes. So not all bacteria are pathogenic. One of the ways to know whether or not the bacteria can cause disease or not is to detect virulence genes. So you can analyze expression of virulence genes in response to the environmental factors, or simply you can use PCR to know whether or not that bacteria has virulence genes or not, and consequently if that bacteria can cause disease or not. And also you can do metabolic and measure metabolic enzymes. For example, if you're studying the effect of growth promoters, drugs, toxins, etc., in response to measure metabolism of certain nutrients, drugs, or toxins in response to feed additives, diet changes, boosters, etc. So you can do gene expression analysis. Okay, so this is just a summary comparing conventional PCR with real-time PCR. So in case you're faced with the decision, should I use conventional PCR or real-time PCR? So for sensitivity, conventional PCR usually has lower sensitivity, and real-time PCR usually has higher sensitivity. So performance, obviously, because you need to do the gel, conventional PCR is longer. The results, you'll only see the results at the end of the run for conventional PCR. Cost is cheaper for conventional rather than real-time. Quantification is relative only to conventional PCR, but if you have standard curves, you can do absolute quantification for real-time PCR. Okay, so summary, PCR and real-time PCR are tools that detect DNA or RNA in a sample, and both are very useful and flexible. But you need to know what you will be using, because each of them has their own pros and cons. Okay, so last slide. If you're planning to do your research, you need to find your objective first. What do you want to do? Okay, so once you answer that objective, you need to do a sample workflow, which is a summary of your method. So, for example, you want to do DNA sequencing, you first proceed to sampling, then sample processing, which you can do a lot. Then you proceed to your first process, which can be a conventional PCR, real-time PCR, ELISA, anything. Then afterwards, you perform DNA sequencing analysis, RFLP, etc. Then go to data analysis. The point of this diagram is there are a lot of combinations of protocols that you can do, and that would depend on your study. What do you want to do? And also on your budget and how much time do you have. So what's important is you know your objective and you know what are the tools and protocols that are available to you in order to achieve your objective. Okay. So that's the end of the lecture. Thank you.
Very much, Ezra. It's jam-packed of heavy technical knowledge. I've been doing PCR myself for so many years, but I don't know the nitty-gritty details, and it's so nice to hear your presentation and to understand better, the details surrounding this polymerase chain reaction. So thank you so much for sharing your expertise and knowledge. So we have a few questions in the chat box. So you may read them and then I will also read them aloud. Let's go to the first one. I think I was the one who first asked this. So what is the main difference in terms of isolation steps in DNA or versus RNA isolation?
So if you're using kits, you will almost certainly you may not be able to see the difference because it's in the buffer. So, but if you look into the theory of the DNA isolation or RNA isolation, for example, if you're using solvent-based methods, one of the steps is to denature. One of the steps is to put the DNA RNA into a buffer. Then what will happen is that you will do, I forgot the procedure, but you will do a procedure wherein the DNA and RNA will be separated based on size, on whether or not they're single-stranded or double-stranded. So the way that you separate DNA and RNA is the fact that most DNA are are double-stranded, and most RNA are single-stranded. So I forgot the specific theory chemical step that they're using, but they use the fact that the DNA is double-stranded and the RNA single-stranded. But when you're doing when you're using kits, you will almost not you will not be able to tell a major difference between the two kinds of isolations.
Thank you.
Doc, point out ko lang din, using a DNA or RNA kit is not a guarantee that you will isolate only DNA and you will also only isolate RNA. So sometimes for DNA isolation kits, you will read in the manual that it might contain RNA, and also some for RNA kits, you will see that it might contain DNA. So it's important to do post-processing, for example, DNA digestion or RNA digestion, if you really want pure DNA or pure RNA.
Okay, thank you for that answer, sir. Another question, what is your favorite brand for the isolation kits and why?
Favorite brand? Wala. I don't have a favorite brand, but there are several factors that you need to consider when choosing a brand. For example, one of the most important is your budget, because some kits are very expensive, some kits are very cheap, but absurdly cheap up to the point that they're not effective. So you also need to be careful. Some have good value for money, which means they're cheap, but they'll do the job. So that's the first one. The second most important, I would say, would be the equipment. Some kits would need, for example, special equipment such as for automated kits, for example, you need the machine, the robotic machines. Sometimes you'll need thermoshaker, which is an orbital shaker and a hot plate. So sometimes you don't have that. For example, in our lab, we don't have that, so you can't use those kits. So another consideration would be the purity or the quality of the DNA or RNA that you need. So some would give a higher, some kits would give a higher purity than some other kits. So usually the ones that are more expensive would, not always, but sometimes those that are more expensive would give higher quality DNA or RNA. So that would really depend on those factors of what brand that you're using. Usually the manuals for these kits are available for free online, so that would I recommend that before you buy any kit for your study, you read on those manuals. They're very long and they're very technical, but instead of spending $500,000 for kits and ending up not using them, just read the manual.
I believe our students also have questions for you. Yes, please take it away, Dr. Racho.
Hi, Sir Ezra.
Hello, Doc.
Actually, nasagot na yung isang question kasi yeah, definitely the budget will also be a factor. And I have the same question with Dr. Lee. Pero dun sa, di ba, pag-choose, you mentioned kanina na yung may mga cheap brands na parang hindi effective. Have you tried this parang, ano mo siya na-evaluate? Or like more on parang reviews lang from your colleagues lang din? Or how would you say na, ay parang cheap to na hindi naman effective? May mga ganong experience. Ano yung mga experiences mo sa ganun?
So usually the only way is to try them.
Yeah.
So to be sure, stay clear from sketchy brands. So I've actually done one evaluation side by side. And hindi naman, it's not always the case though, but I've tried a no-name brand versus a known brand of RNA Isolation kit. I won't mention the brand, but when I compare them side by side, the brightness of the commercial kit that is well known is higher than the ones that is not well known. So it may or may not matter. For example, it could matter if you're doing diagnostics, clinical diagnostics, for example. In most cases, you may want to settle for the well-known or branded kits. But if you're doing cultivation where you have a lot of starting material, for example, material culture, then it's possible to settle for the less expensive kits. So, but personally, I play safe. So one of the good ways to determine whether a kit is usable or not is to look at the publications. So usually they'll give their the kits that they're used. So you'll see usually the brands are declared. So hindi naman, it doesn't mean that you have to stick to US made or to Europe made. There are also a lot of kits from Asia that have been used in publication. So read the publications on your target journals and you can narrow down the list of kits that you will use.
Okay, pahabol lang ako. Kasi
nga pala, Doc, tama yung sinabi mo with regards to the market. I forgot, it's also important. Yeah, what's available because not all kits are available.
Yeah, especially kung madaling kunin yung acquisition procurement. Doc, then habol lang ako ng follow. Ayun, kasi na-mention Doc kanina, the brightness, related kasi dun sa parang color yung tanong ko for the RT-PCR that you mentioned, di ba, yung multi-probe. Na you mentioned 5 colors kanina. But you mentioned before that na parang may have a journal na nakita na they use up to 9. So parang feeling ko kasi parang kung 9 colors, parang siyang, parang ang hirap i-differentiate minsan ng ibang shade. So, di ba, parang paano kaya yung mga gano'n na color?
Usually kasi predetermined yung dyes. So walang ano yan, walang light red, walang red, walang ganon. So for example, you'll use for example FAM for green, sci-fi for red. So very specific dyes. You won't use colors really, but you'll use channels. So each channel, each dye belongs to a different channel. So if I'm not mistaken, there are 5 channels for the visible light spectrum. So they are red, orange, yellow, green, and blue, if I'm not mistaken. So you'll only use the five channels for the visible spectrum. So the machine will not be confused because each dye is dedicated only for one channel. So you cannot use two dyes that belong to the same channel. So for example, if you use, I think another red dye is rhodamine, CHR, then Cy5, you cannot use both dyes in a reaction because they belong to the same channel. And the PCR machine will not let you.
Yeah, yun nga. Thank you, sir.
Welcome.
We have our next question. Doc Jen, please take it away.
Hello, sir. Good afternoon po.
Hello, good afternoon.
Ayan. Based po dun sa experience niyo, sir, what are the problems that you have encountered using yung PCR po?
Conventional?
Real time.
Real time, one of the biggest problems would be what we call PCR inhibition. So this is very prevalent for samples that we consider to have a high levels of interfering substances. For example, blood, common culprit, and fecal matter, common culprit din yan. So what you will see, a lot of the real-time PCR reactions have two signals. So you would have the signal that is always positive and also you have the target signal. So yun yung tinatawag namin internal control. So if the internal control is negative, then it means you have PCR inhibition. So ibig sabihin nun, mali yung reaction. False negative, likely. So we usually encounter that when we do real-time PCR with fecal matter. So the solution to that is sometimes they don't follow instructions on the kit. So very, very, very common, unfortunately. So usually they don't perform the last centrifugation step. So yun, nagne-negative, yung false negative reaction nila. Then another solution to that is to dilute your DNA. Because real-time PCR is very sensitive. So it sometimes, even if you have very, very small amount of DNA, you can still detect it. But in diluting, you also dilute the contaminants. So you decrease the number of DNA, which is still detectable, no problem, but you also decrease the number of contaminants, and therefore your PCR reaction will likely succeed. And also primer specificity, very common and also a personal problem of mine. Once I order a primer which is used in a journal. So proven primer that works. It took me I spent 3 weeks trying to get it positive using optimization, but it doesn't work. So what I did is I ordered another primer for the same strain but different location on the genome, and on my first reaction it worked. So as my advisor mentioned, primers are very finicky. They are weird substances. Sometimes they work, sometimes they don't work. So my advice would be, if your primer doesn't work and you're pressed for time, it might be a good decision to find another primer, or you can design your own primer if you want. Yeah. So usually those two are the most common. There are a lot of problems, but personally, very common.
Yes, very well answered. Thank you. Doc Jen, you still have your last question?
Yes. Sir, how long po will it take for one testing po using the real-time PCR? Since for the regimens po, yung during the diagnosis ng disease natin. So, we need to give specific medications dun sa patients natin. So, how long will it take po for one cycle? Is it cycle yung tawag po ba?
One run.
One run.
Yeah. So it will depend on whether or not. So number one, it will depend on whether or not you're detecting for RNA or DNA. RNA is usually longer because, for example, if you're detecting PED, for example, it will take longer time because it usually you will have to add 30 to 60 minutes of reverse transcription. So if you remember sa lecture kanina, you cannot do, or at least as far as I know of, you cannot do PCR on DNA. So you have to convert RNA first to DNA, and that would take time. So in my experience, personal experience, it can take anywhere between 1 hour 45 minutes to 2.5 hours, depending on also on the number of cycles. Of course, if you also only use 25 cycles, that's fast. If you use 40 cycles, that's long. But there are protocols for DNA that are termed fast protocols. If you use fast reagents, they say, I've not tried this, but they say that you can do PCR, real-time PCR, as short as 15 to 20 minutes. So I've not tried it personally, but theoretically you can do 20 minutes. But usually, by experience, it takes 1.5 hours to 3 hours at most. Conventional longer can take you 5 hours.
Yes, we have a follow-up question from Dr. Rio.
Actually, hindi siya
Hindi pala ako nagmamadali, Doc.
Hindi naman siya actually follow-up. Naisip ko lang kung if you're familiar, kasi in small animal practice, so hindi rin ako familiar pa dun sa machine. I've heard about in-house mga PCRs sa mga clinics. Are you familiar with this? Tapos ano yung thoughts mo? Kasi ako personally, I haven't seen it yet then. Pero I've heard na parang hours na lang, tapos in-house, kaya siya.
Yes.
Have you heard about this?
Paano in-house, Doc?
Ano siya? Actually, Doc, I think you're familiar also with this, no?
Yes.
Parang nakakit na Doc, mukhang kailangan kong i-share mo, pakita na lang sa'yo next time, Sir Ezra, yung parang manual niya. Kasi hindi ko nakikita yung, kasi parang I don't see yung usual setup na ginagawa sa parang dyan, sa microbio, small bio, dun sa mga in-house sa small clinic. Para siyang small machine lang eh. So, if ever kasi you're familiar with that, I wanted to hear your thoughts. Pero siguro, I'll share it with you na lang, Sir Ezra, in the next time na. Tapos, pakita ko sa'yo doon kung anong thoughts ko. Kasi
sige, Doc. Sige, ikaw muna.
Dahil na, Doc, hindi ako ganun din familiar with the setup. Tapos parang, hindi naman na doubtful ako, pero I haven't really tried it yet. So parang, mas gusto ko pa din ipadala na lang sa mall bio ang mga tests rather than in-house na gagawin siya. In-house siya mismo sa clinic eh.
Actually, the reason why I asked, Doc, kasi I'm familiar with what are called point-of-care diagnostics. I'm not sure if you've heard of it, but the point of care diagnostics is you go to the place where the patient is, example, farm, point of care, farm, hospital, etc. Even though they don't have a dedicated laboratory, you can just bring the equipment, do the diagnostic, get out. That's the point of. Well, we actually have one in the mall bio. It's called the Pocket PCR. I'm not sure if you're familiar with it. It's also a point of care PCR. So that one is OIE approved. So it's unlike the PCR that we do in research, it's actually diagnostics rated. So you can actually use this for patients. So yes, those exist. Merong mga tinatawag na point of care molecular diagnostic kits such as the Pocket kits.
Usually, Doc, ganun din siya katagal. Ganun din siya katagal usually.
Since nucleic acid based siya, it would take 2 hours. From isolation to detection, usually 3 hours. So we've done it in the field. We've tested in the field and it works. So advantage niya is portability. So yun yung main advantage niya. Actually, kahit even sa COVID, you can see that usually, not always, the PCR-based methods are take longer time. Kaya nga na-favor yung antigen-based, di ba? Kasi they take shorter, sometimes as short as 15 to 20 minutes. But yes, those exist. And my suggestion is, if you're using it for diagnostics, is you do an in-house verification, or you check whether or not they have approval of a regulatory agency. So for example, I'm not sure if DA approves diagnostic kits for, at least for the PAC, if they are OIE approved. So yun, they exist. They do exist.
Okay, sir, thank you so much. We really learned a lot. No, coming from me, I really learned a lot. And taking also your personal experiences in doing this type of research. Okay, let's give Sir Ezra another round of applause. Okay. Okay, and to follow po ang aming certificate of appreciation to you, and we don't want to extend more of everybody's time. We are now 2:11. Okay, so in a bit, I will be ending this session. Once again, thank you so much, sir. Thank you. For my class,
Thank you.
next Thursday again. Bye.
Bye po. Thank you po,
Bye
sir.
po. Welcome.
Thank you
Thank you
po.
din.